I'm Bob Levinson from the Institute of Personality and Social Research and the Department of Psychology. And I'm going to serve as sort of the host for the first day. And then my partner in crime, Ron Dahl. Ron, you're here, right? I heard a rumor you were here, but I don't see you yet. Yeah, there was a Ron sighting. He's going to serve in this role tomorrow. So welcome to everybody. And I'm going to tell you a little bit in these opening remarks about how we came to be here today, and what we're up to in the long run, and what our goals are for the conference today. So as you probably know, there's Ron. Yay! So that's Ron. This is Bob. You can always tell the difference. He always has the knapsack, and other than that, we're pretty much the same. And so if we had this conference a decade ago, we would probably be, and we were interested in goals of well-being and health, we'd be probably talking about microbes and bacteria and the immune system. And for decades, that was the focus of the National Institute of Health. And then somebody had the great idea, why don't we kind of look at the burden of illness and see whether or not really we're looking in the right place. And sure enough, when you look at the burden of illness, you find that behavior makes this huge contribution, not only to things like depression, which is one of the major economic health costs in the country, but also involved in all sorts of health promoting and risky behaviors that increase the risk for other things. And these great public health initiatives that are part of our lifetime, like getting people to buckle up and stop smoking, have had huge impact. And in fact, in some ways, some people who have said that there's been more traction from behavioral interventions for improving the nation's public health than in a lot of the more basic biomedical research. So seeing this as an opportunity, we don't have a medical school at Berkeley, as you know. So our lack of a medical school all of a sudden became an advantage. A group of us started meeting in 2011 to talk about whether or not Berkeley could be part of this growing initiative. And we formed something called the Behavior Change Research Network. And we had a couple of ideas that I think were a little different. One, I give major credit to Ron for, and the idea was that if you're thinking about changing behavior, there probably are a few key points in the lifespan where it's particularly important to intervene. And this is this idea of inflection points. And these are times in our lives when things change in our biology, in our social worlds, in our motives and values, and we have a chance to sort of reset or reboot and set a trajectory that's going to influence the way our lives unfold in coming decades. And perversely, we'd thought that rather than studying early childhood, the typical kinds of times, that we would focus on adolescence and the beginning of late life. These are times when people's motives change, the social world becomes really important, changes occur, we'll hear some of this today, the brain ages and develops in interesting ways, the frontal lobes change both dramatically in adolescence and in early late life. We get better at some cognitive things and worse at other things. Emotions start to become a very important determinant of the things we do. We make critical decisions that are going to influence us and the people around us. So we thought there might be some gain to be had, some traction, by studying these two points of development in concert. So that was one of our first ideas. We also decided that targets for some good targets for interventions for behavior change might be the way we regulate our emotions, our motives, and the way we make decisions. And again, these are things that undergo interesting changes at these points of the lifespan. The nature of this effort was, from the beginning, going to be multidisciplinary. And we've had the participation of people from business, demography, economics, psychology, public health, and sociology, also neuroscience. And as going forward, and we're hoping to go forward, we expect that we will be making further outreach. We have some people in engineering who we've been talking to, and trying to put together a kind of infrastructure here at Berkeley that will eventually foster and foment a new kind of interdisciplinary research on behavior change. Our activities in the first 2 years have been retreats. We're now sponsoring our second interdisciplinary seminar. The first one, which we sponsored a couple of years ago, was the only graduate seminar in my many years of experience that had a waiting list. And that wasn't auditors. People actually took it. It was a multidisciplinary seminar. We are trying to put together interdisciplinary research teams. We've had a stream of spectacular postdocs over the past 2 years who have been part of this program, and we've had conferences, and that's where we are today. One of the first things we did is we got a conference grant from the National Institute on Aging, and it was a funding mechanism which was designed to create interdisciplinary research teams at a particular site. And so the goal of these conferences are to bring in the best thinkers, the best scientists, the people who are trying interventions, developed interesting interventions in the realm of behavior change, to sort of talk to us, so we can learn about their ideas and their approaches, get their reactions to what we're thinking about, and really help us in this process of building our research network here at Berkeley. This is our second conference. We had the first one last year. Maybe a slightly more basic science approach. Both were designed to be a mix. This one may be more of a focus. You'll be hearing about a lot of really interesting interventions over the course of the 2 days. And then as part of the conference grant, we've committed to a dissemination effort that includes conference reports, and we have a science writer, Sandy. Sandy, want to stand up so people will see you? Sandy will be taking notes, and she'll be producing a serious document at the end of this that will be available to everyone on the website. We also are having all the talks videotaped by our ETS group at Berkeley. This is, I think, the third or fourth time we've worked together, and they do a great job. And so there'll be first-rate videos available on the web rather quickly. The conference format is pretty simple. This one, we simplified it even more. The last one was supposed to be simple. This one's even simpler. The highlight is 7 spectacular thinkers in this area. Each will be giving a talk, 4 today, 3 tomorrow. If I've misspelled anyone's name, please forgive me. I tried really hard, but I actually did this myself with my own little stylus last night. So these are our speakers. You'll see they come from all over the country. They come from a number of different fields. And as you'll see today, they're all doing spectacular work in the area of behavior change. After each talk, there'll be chance for discussion, questions, and answers. One of our BCRN postdocs will be hosting each of these sessions. And then at the end of the day, both days, we'll gather together for a brainstorming session, trying to extract common themes and selfishly using these wonderful resources to help think about the future of this project at Berkeley going forward. The other thing we are doing is we've built in a lot of social times. So you'll see lots of food, mostly from Rick and Ann's or Ann's Catering, which is actually quite good. Breakfast every day. We're bringing in lunch. There'll be coffee breaks. And tonight we'll have the only thing that isn't a public event. The core executive committee of the BCRN will be having a dinner with the invited speakers. But other than that, everybody's welcome to everything and eat a lot of food. And, at the end, we'll give out plastic bags. This thing turned out to be very complicated to put on. no sooner had we gotten the grant where NIH changed their policy about food, and we found out that we had all these conferences planned, and we couldn't even serve you coffee. So we went to a round, and we got a lot of additional support from the campus. So if you're enjoying the catering and you're enjoying your box lunch, there are lots of people to thank for it. So the National Institute of Aging gave us the main conference grant, and our Dean of Social Science Division, Carla Hess, and her Associate Dean, Catherine Carson, who will be coming by later, and I'll introduce her. They have been supporting us for 2 years, helping us with the postdoc support, so that's been terrific. All of these other institutes are on the Berkeley campus. They're mostly research institutes that have sort of invested in this program. So I'm going to mention them quickly. The Berkeley Population Center, the Center on Economics and Demography of Aging, Experimental Social Science Laboratory, the Institute of Business and Economic Research, Institute of Human Development, Institute of Personality and Social Research, and the Robert Wood Johnson Foundation. Also want to thank our staff. If you've had any dealings with Elizabeth Peel, who's standing in the back, looking incredibly calm, considering, how many plates are up in the air. But Elizabeth has been the major mover in enabling us to organize this conference. Lisa Branham from IHD, I think, is out at the front desk and around the corner. And Sandy I've already introduced. We have a group of assistants. I'm told that every member of my lab volunteered to be an assistant. So we'll make a slide up and we'll thank them a little later. They're not all here yet. I also want to thank the BCRN faculty, in particular Ron, Shahar Karif, whose mother was hospitalized last night and may not be able to attend, Ray Catalano in public health, Will Dow in demography, Bob Bardet, who has been, really was the guy who got us going. Anybody else should thank? You probably weren't following that closely. It's kind of early in the morning. And whoever I forgot, my agent, the people at Fox Searchlight for greenlighting this project, and of course Sally Fields for teaching us all how to say thank you. These are the postdocs who have worked with this program. It's really an amazing group. We have 2 generations, one that's gone on, and now many of them have on their first faculty jobs. And Anna and Nana, it's kind of funny, are both here this year as our new postdocs. And then we particularly want to thank our speakers and our attendees. Oh, that's great. I'm going to be on time. So, it turns out there's a lot of controversy over whether it's ADIEU, A-D-I-E-U, or ADO, A-D-O, I guess, ADO. How many of you think it should be ADO? And it turns out, that's what I thought too. And Emily, if you would know this, because you're the sort of the English scholar. But it turns out this is what it is. And it means without further hubbub and mess. And it comes from Shakespeare. So as the purveyor of mess and hubbub, I'm going to pass the baton here to Jeff Spielberg, who's going to introduce our first speaker. And thank you all for coming.
So thanks, Bob, for that great intro. And while Adriana sets up, I just want to introduce her. I'm very excited for our first speaker, Adriana Galvan. She's an assistant professor of developmental psychology at UCLA, and she's going to talk to us today about adaptive risk-taking in adolescence, insights from the developing brain. And just one correction for Bob, who I'm sure was very careful but missed one thing, is that we are not going to have discussion after each one. We're just going to have questions, and we're going to try to save the discussion for the end of the day to try to interweave all the talks of the speakers. So.
I think so, yeah. Hopefully it's going to work. Okay, great. Hello, thank you so much for inviting me. And Bob, that was such a wonderful overview of this amazing network. I wish I could be a part of it too. It sounds wonderful and so interdisciplinary. And I hope that my talk is relevant, and I think it'll be more on the basic neuroscience side of things. So it sounds like we'll be hearing a lot more about interventions and manipulations. And in my laboratory, we're interested in studying naturally occurring or perhaps naturalistic behavior change that is inherent to the adolescent window of development. And what we do is study the developing brain, and from all of the research we know from animal models, that there are a lot of ontogenetic changes in particular brain networks. We're really interested in understanding how those structural and functional changes in the brain help us become adults. How is it that we become adults across this really critical period of development? And because a lot of behaviors that emerge during adolescence are often associated with changes in how we make decisions or the types of environmental information that adolescents are attending to, I think a lot of media focus, or maybe layperson focus, is really on risky decision-making during adolescence. And of course, adolescence is a lot more than just risk-taking. But we ask whether or how risky decision-making facilitates autonomy or becoming an independent person. So I'll review in brief several years and research from a variety of different labs showing that the adolescent brain is special, and I think that's something that we now all can agree on. Every few years, it seems, there's some national or big magazine covers the teenage brain or the mysteries of the teenage brain. And that's in part because brain imaging has become more sophisticated, and many labs are using it to uncover changes or characterize changes in different networks of the brain. So beginning in childhood and continuing through adolescence, dynamic processes drive brain development, and they create the flexibility that allows the brain to refine itself, to specialize, and to respond to specific demands of the changing social landscape. And these maturing connections pave the way for increased communication between disparate brain systems. Today, I'll mostly focus on systems in the prefrontal cortex and those in more affective regions, including the ventral striatum. And so during these changes, when what we call adolescence arrives, there's a changing balance, or there's hypothesized to be a changing balance between brain systems involved in emotion and reward sensitivity, and those that regulate emotion or affect. And presumably this spawns the increased exploration, the willingness to take risks, and simply the behavioral change that is often observed or characterizes adolescence. And these behaviors are remarkably found in all social animals. So it's not just in humans, but in non-human primates and rodents. And they seem to facilitate these important developmental milestones that pave the way for new adventures that are at the very core of transitioning from a state of dependence to one of relative independence. So my laboratory is really interested in how the specialness or the uniqueness of the adolescent brain may help us transition into adulthood. So the unique changes in the brain are centered around a few things. One is that the adolescent brain is malleable or adaptable. And of course, all of our brains are, right? We're constantly responding to the environment. But there seems to be a particularly unique malleability and responsiveness to the environment during adolescence in many domains. And perhaps this is meant to optimize or to facilitate independence. So how does the brain do this? Or what happens? This is a figure that many of you have probably seen before. It's from Elizabeth Sowell's work in 1999, showing that there are significant structural changes in the brain. And this is the front of the brain, this is the back of the brain. And what it's showing here is that the regions that are in different colors, so in the purple, this is overlapping the areas of the prefrontal cortex. The green is representing the striatum. And these colors are meant to represent regions that change most significantly between adolescence and adulthood. And what Elizabeth found is that there are significant decreases in volume, which is for large body of literature I won't review, thought to be an index of maturation of the brain. And interestingly, these same systems, the striatum and the prefrontal cortex, are critically implicated in decision making or in making choices that might modify behavior. What's really interesting about the relationship between these two systems is that it changes dynamically across the transition into and out of adolescence. So in pre-adolescent humans, there seems to be a rather tenuous relationship between the prefrontal cortex and what I'm focusing on here, which is the ventral striatum, the dopamine-rich ventral striatum. And of course, these systems are talking to each other, but the connectivity between the two seems to be in a rather immature state in childhood. In adulthood, a large body of research suggests that the prefrontal cortex has become central to regulating behavior and to responding to making decisions or representing decision making, and that the prefrontal cortex exerts a rather strong influence over the ventral striatum that perhaps helps regulate affective responding. But that during adolescence, there's strong sensitivity in the ventral striatum. Most of the research has focused from the brain side on reward sensitivity in the ventral striatum, and I'll review some of those data. And that this heightened sensitivity exerts a bias over the prefrontal cortex, which is still in a rather immature state. And this is a very simplistic view, and I completely appreciate the fact that both of these systems are talking to one another. But the critical point here is that the ventral striatum is very responsive to the environment. So why is that? Well, the ventral striatum is rich in the neurotransmitter dopamine. And dopamine perhaps is one of the key signals that helps us transition into a state of relative autonomy, or to help facilitate this transition. And I thought it would be worth reviewing what we know about dopamine and about how that system changes in adolescence. So here's a schematic of dopaminergic projections. And dopamine neurons originate in the midbrain and the substantia nigra in the ventral tegmental area. And they project up into the striatum primarily, and then also into the frontal lobes. And just want to remind you that these are also the systems that change significantly during adolescence, or from adolescence into adulthood. And from, of course, in humans, we can't really test how dopamine firing and dopamine receptor availability is changing, but we can examine these data in rodents, which has given us a lot of insight. And these data suggest that dopamine neurons and dopamine activity really changes during adolescence. So this is a cartoon of a hypothetical dopamine neuron. And if this synaptic vesicle is releasing dopamine into the synapse and binding to receptors, data from a lot of work from Susan Anderson's lab and Linda Spear's group has shown that during adolescence, there's not only an increase in the amount of dopamine available in the presynaptic neuron, but that there's greater dopamine release in response to similar stimuli. So in response to a reward, the adolescent brain shows greater dopamine release, more firing rates, and greater receptor availability. And like I said, we can't directly examine this in humans, but what we can do is use functional imaging as an index of where we believe these dopamine neurons are acting, which is in the striatum, in the ventral striatum, which is what I'll focus on. Okay, so let me just. Okay, so dopamine, a lot of the literature has really focused on reward sensitivity and the role that dopamine plays there. But dopamine, first and foremost, is really a learning signal. And it's meant to learn about the environment and to make associations and generalize across different contexts. It's also critically involved in valuation, exploration, adapting to the environment, and it has a role in increasing social interactions or interest in peer affiliation. And so I'll review some studies that suggest that there are unique changes in dopaminergic systems or regions rich in dopamine in all of these different constructs that may help facilitate this overall picture of autonomy or independence. So the first few studies I did in this area were to examine whether or not there were ontogenetic differences or changes in striatal systems across the transition into and out of adolescence. And so what we did is we brought a group of kids, a group of teenagers, and a group of adults into the laboratory and presented them with different rewards. In this case, there were monetary rewards. And what we found is that all age groups, as predicted, showed robust activation in the ventral striatum in response to simple monetary rewards. But when we pulled out the signal for each group, there were clear developmental differences such that there was this inverted U-shaped development with exaggerated activation in the adolescent group in response to the very same reward. And so this was just one bit of evidence, and other laboratories showed similar findings using other types of tasks and whatnot. But we also wanted to know whether this was, we wondered if there was a confound here, because using monetary rewards with adolescents may be confounded with the experience of money, right? Adolescents have less experience with money, and so perhaps that's inflating their response to monetary rewards. And so what we did is that we borrowed a paradigm from the non-human primate work in which monkeys are trained to learn that a cue predicts a particular reward outcome, and that reward outcome is a drop of juice. And so rather than using, of course, secondary rewards like money, they use primary rewards like juice. So that's what that setup looks like in a monkey. They are trained that this cue predicts a drop of juice, and the juice is delivered through straws. So we did the same thing with humans. We brought the kids into the lab and we set them up with straws in their mouth. It wasn't trivial, but it's actually fine. And they drank sugar water, salt water, and regular water. And of course, we ensured that they found the sugar water pleasant and all of those types of controls. But the punchline is that I'm sorry, I said children. We didn't have a group of children here, just adolescents. The punchline is that in response to the pleasant taste or the rewarding liquid, the adolescents showed more robust activation relative to the adults, which lends further support for this idea that the type of reward is less relevant, and what's relevant is the sensitivity of the striatum to what's perceived as a rewarding stimulus. And this was associated, we asked them after each trial how rewarding they found the event, the stimulus. And in the adolescent group, those who rated it more favorably showed a parallel representation in the striatum during the sugar water delivery. And this association we didn't find it in adults. Okay, so one more bit of evidence to test whether the type of reward is critical in showing this unique response in the adolescent brain. And to this end, instead of delivering rewards to the adolescent, we had them complete a task in which they gave money to somebody else. And we were interested in whether or not that elicited this similar robust activation in this region. And so participants came to the lab and played a charitable donation game in the scanner. And in this case, they were giving money to their family member. So it was called a costly donation because it cost them money to give money to someone else, because that money wasn't something they were going to receive in their pot. And what we find is, again, that even more robustly, that the costly donations elicited activation in the striatal region when given to their family. And this is work done by Eva Telzer, who was a graduate student with me a few years ago. Okay, so this is, for us, convincing that reward sensitivity during adolescence is represented in the striatum. But the subsequent question that we had was whether or not this heightened striatal sensitivity is functionally meaningful. What does it do for the adolescent in terms of either changing their behavior or serving an advantageous purpose? So we asked whether heightened striatal sensitivity enhances learning during adolescence, and whether it enhances valuation in a way that promotes optimal decision making. So to address the first question, we collaborated with Daphna Shohami at Columbia, and she's really interested in basic learning systems. And what she's shown is that the striatum is very responsive to immediate feedback. So when it's learning about feedback, when it's learning and the feedback is immediate, it's represented in the striatal system. So we thought that that would be a really good place to start because we can use a task that we know probes the striatum. And based on her previous work, we predicted that immediate learning as opposed to, or immediate feedback as opposed to delayed feedback, which will make sense in a second when I describe the task, would engage the striatum as opposed to the delayed feedback, which would engage the hippocampus. So this is the task. This does not involve reward. It simply involves feedback. And we purposely didn't introduce reward to the task because we wanted to isolate the effects of the reward from the effects of learning. So in this task, participants are presented with one of 4 butterflies, different colors. And each butterfly, and they have to guess whether the butterfly is associated with one of 2 flowers, yellow or pink in this case. And each butterfly is associated with each flower either 20% or 80% of the time. So for instance, this orange butterfly, on 80% of the trials, the correct answer is pink. Okay, does that make sense, the probabilistic nature of the task? Okay. So then after a 1-second delay in the immediate feedback condition, they receive a feedback that says correct or incorrect. And when they receive the feedback, there is a seemingly irrelevant image associated with that feedback, and that'll become important later. So this is the immediate condition. In the delayed condition, it's the same task, except now, following the response, there's a delay of 7 seconds before they receive the feedback. And in the scanner, that's a really long time to wait because you don't know right away if you made the correct choice. So these are the behavioral data, and what we find is that with the optimal choice here on the y-axis, that the adults show incremental changes across the task. So block 1 is the beginning of the experiment, block 4 is the end. And there were no differences in their learning between immediate and delayed feedback. In the adolescents, there were also no differences, but what we did find is that they showed a steeper slope or a steeper increase in the learning rate between the beginning and the end of the experiment. And contrary to what we predicted that the immediate feedback would show greater learning, but it didn't. However, what's interesting to us about these data is that in virtually all studies of memory or learning, adults outperform adolescents, whereas here we saw the opposite, where the adolescents were showing better learning overall than the adult group. And then once outside the scanner, about 30 minutes after they completed the task, the participants were given a surprise memory test. And the memory test was one in which we presented the images that had been associated with that feedback screen, so that picture of the phone or the picture of the beach ball. And we asked them whether they were images that they had seen previously or whether these were new images, the foils. And what we find is that in the delayed condition, the images, there were no differences between adolescents and adults in which images they remembered. But in the immediate condition, in other words, when the feedback came right after they made their choice, the adolescents showed significantly improved memory recall for these items compared to the adults, suggesting that there was greater learning, or at least encoding, in response to immediate feedback. And remember, the immediate feedback, based on Daphna's previous work, is rooted in the ventral striatum. So we did an imaging analysis to test this very question, and what we find is that this learning rate, or this learning signal, was differentiated in the brain, and the place where it was differentiated in the brain was, as you might guess, I wouldn't be telling you, was in the ventral striatum. And for those of you who do imaging, this looks like a region of interest analysis, but it's really the only brain region that popped up, which is really remarkable to see that it was definitely isolated to this region. Okay, so these data suggest that learning, or enhanced learning, in the adolescent brain seems to be supported by changes or differences in the ventral striatum between adolescents and adults. Okay, so the second question about whether this heightened sensitivity is functionally meaningful was one we asked by asking whether the adolescent brain was valuing available options in a different way. And so to do this, a graduate student in the lab who recently graduated, Emily Barclay Levinson, conducted an imaging study in which she used a task that presented gambles to the participants. And these gambles were such that we could calculate whether it was a good gamble or a bad gamble, whether it was advantageous or not. The task is one that we borrowed from Craig Fox and Russ Poldrack's paper in 2007. And it's a very simple task. So they're presented with this pie chart, which is a gamble. And they make a choice of whether they will accept this gamble or reject it. And the gamble isn't resolved during the experiment, but they know that we will choose at random one of the many gambles that they've done and play it for real money. So in this case, they're choosing whether they will accept the gamble of a 50% chance of winning $5 or of losing $10. And the way we calculate whether this is a good gamble or not, or an advantageous gamble or not, is that we calculate the expected value of the gamble. And that's basically, for those of you who are unfamiliar with it, the product of the monetary amount by the probability of receiving that, or of getting that gamble. So the values were sampled from a matrix. So the potential gains ranged from $5 to $20, and the potential losses ranged from minus $5 to minus $20. And so, for instance, this gamble here on the upper right corner is showing you a gamble that has a positive expected value of 6. So in other words, you should accept that gamble because it's a good one. You have a 50% chance of winning $17 and compared to a 50% chance of losing $5. But there are also gambles in there that had a negative expected value. So for this gamble, the answer should be reject. They're not going to take it. And then we also threw in some neutral expected value trials where there was no better or worse choice to make. Okay, so these are the scanning parameters for those who are interested. And what we find is that the manipulation was effective in that participants in both groups were more likely to accept the positive expected value trials relative to the negative expected value trials. So they knew which ones were better. They were able to differentiate them. And importantly, there were no group differences. So the adolescents did not differ from the adults. In other words, the adolescents were just as good at picking up on the advantageous trials and making choices that were appropriate for the gamble that was presented. There were also no differences in groups between the neural systems that supported representation of increasing and decreasing expected value. So regions in the frontal cortex were representing increasing expected value, while regions in amygdala and insula were representing values that were decreasing over, across the experiment. But what we wanted to know was whether there were significant effects of the slope of the EV. In other words, as the expected value is increasing or decreasing, were there differences in how adolescents and adults were responding to them? And we did HLM to test this question. So here I'm showing you the mean response on the y-axis. 1 means they accepted it every single time. 0 means they didn't. And the expected value is here on the x-axis. So everything in red here are the negative expected value trials. They shouldn't accept the gamble. And then the green represent the ones that they should accept. So there were no differences, virtually no differences at all, between behavior that the adolescents compared to the adults did on the neutral or the negative expected value trials, as the main effects suggested. But where the differences emerged and where there was a significant effect of the slope of EV on their response is on these increasingly more positive or more advantageous trials. So as adolescents were presented with trials in which they were the most advantageous, they were actually showing more optimal behavior compared to the adults. And of course, we wanted to know where there was a neurobiological representation of this. And so we did, and we had our hypothesis about the ventral striatum, so we did an ROI analysis. And indeed find that this greater sensitivity to the increasing expected value, which leads to the more advantageous choice, was represented in the ventral striatum more robustly in adolescents compared to adults. But to isolate the unique contribution of ontogeny to the observed neural differences between age groups, we conducted a secondary analysis where we matched participants based on their own behavior. In other words, if we took the adults who looked or who performed like adolescents, would they show the same neural representation as the teens would? Is this simply an effect of the fact that they were choosing the highest expected value trials more? So we mapped out our data, and here you can see there's nice variability in both the teens and the adults. And we focused on all of the participants who were more likely to accept the gamble for the most advantageous gambles in both the teenage group and in the adults. And what we find is that even when we control for behavior, so when we match the participants for behavior, the adolescents in the show greater activation in the right ventral striatum at threshold. This is a subthreshold on the left. But nonetheless, what you can see is that the exaggerated activation in the teenagers is to potential reward it holds even when we control for the behavior of the participants. Okay, so that's some evidence to suggest that how the teenage brain is valuing available rewards or risks in the environment is mediated by these dopamine-rich regions or supported by them, and may lead to behavior that's more advantageous in this context. Relatedly, we were interested in the increased exploratory behavior that's observed during adolescence, and wondered whether these same systems support exploratory behavior when it's advantageous to be exploratory, to exhibit more exploration. So what we did is we brought in a larger group of participants, and for anybody who's interested in the methods, they're listed there as well. But what we did is we asked them to perform a task that I know isn't perfect. We can certainly talk about that later. But we used the balloon analog risk task, and probably most of you know this task, but just to remind you, what happens is that the participant is presented with a computerized balloon, and they are given the option to inflate the balloon. When they inflate it, they earn 25 cents on each pump. But at any point during the trial, the balloon could explode, and then they lose their earnings for that trial. On the other hand, they could keep pumping and then choose to cash out to avoid the potential punishment or loss of the earnings. And so this task was originally designed to mimic the dynamic nature of making choices in the moment, of taking the next risk or pulling back a little bit in order to avoid punishment. And we created a version of the task that really favored pumping the balloon that really favored maximum risk-taking. In this case, this pumping is an index of risk-taking, or thought to be. And also that maximized so that maximum riskiness on the task earned the participants more money. And we didn't tell them this explicitly, but they were to learn this across the time on the task. Another thing we were interested in is whether or not there were differences in how exploratory people were. So the nice thing about the task is that people really, there's a lot of variability in how people approach this task. Some people really play it, very, very conservatively and always cash out early on. Whereas other people, pump up until the balloon explodes and then they learn to scale back that way. Others really do a range of both of those things. And we were wondering if this exploratory type of approach was more beneficial. And again, beneficial here at advantageous is indexed by how much money they earned on the task, which they got to keep at the end. So what we find is that, indeed, for both of these variables, maximum pumping on the task and this exploratory behavior, individuals who did both of these things did these things to a greater extent earn more money on the task overall. And because we had a healthy age group or sample size, we wanted to know that whether variability in the maturation of the systems that we think are implicated in advantageous decision making might tell us a little bit more about the variability in the behavior. And so to do that, we use DTI, which is a nice way to examine anatomical integration of disparate brain regions, in this case, frontal systems and these limbic regions. And the reason it's a nice approach is because DTI, and based on a lot of different work, DTI has allowed us to map out different tracks in the brain that connect different parts of the brain. And these tracks don't develop along the same time scale. Some show a more protracted development. So for instance, this graph is showing that the uncinate vesiculus continues to mature, or the integration between these pathways continues through the teenage years and into early adulthood. So this tract is more dynamic, and there's necessarily going to be more individual differences in maturation of that tract across the period of adolescence, as opposed to other tracts like, the ILF, which is pretty much mature earlier in life. And our dependent measure was something called fractional anisotropy, which is considered an index of the maturation of these tracts. And what we find is that, so now that y-axis is what representing the dependent behavioral variables of maximum pumping and exploratory behavior. And what we find is that greater fractional anisotropy, which again is considered to be an index of the maturation of the tract, is associated with this maximum riskiness and this exploratory behavior, which, remember, in this task was designed to be the advantageous thing to do. And we find the same pattern in this uncinate vesiculus region that shows this wide variability across adolescence. So these data suggest that exploratory behavior, or exploration in a context in which it's advantageous to do so, is supported by the integration of pathways between frontal and these more affective regions. Okay, the last thing I'll talk about in the last few minutes is this idea of adapting or responding to the environment. And of course, within an fMRI context, it's really challenging to do to change the landscape within one experiment. So instead, what we do is that we've examined teenagers in either naturalistic changes in context or their own inherent differences and variability within groups. And I won't go into the details too much of each of these experiments, but we've tested differences in how changes of basic needs like sleep influence differences in striatal sensitivity. Stress is another one, and then social relationships within the family context. So first, sleep, and this is something that was really, my interest in sleep was really inspired by Ron, a million years ago when I was, still like a first year in graduate school. And I've now recently gotten the opportunity to really start examining sleep. And the first study we did yielded some interesting results in that we didn't manipulate anything. We simply noted differences in sleep patterns, and then asked participants to perform the BART task and determine whether there were differences, again, in this functional connectivity between these regulatory and these affective systems. And what we find is that poor sleep quality was associated with functional coupling between the ventral striatum and the dorsolateral prefrontal cortex, such that participants who reported poorer sleep quality for whatever reason were less likely to have good communication between these systems. And as I said in the beginning of the talk, a hallmark of what we consider a hallmark of mature adult brain systems is this really nice crosstalk between these systems. So if during adolescence these systems are already tenuous and dynamic, something like sleep might further exacerbate them. But of course, we don't know if it's the chicken or the egg. Is it the sleep causing it, or is it the fact that the integrity of these systems predisposes particular behaviors? We also examined whether there were neural differences based on arousing context. So in this more involved study, what we did is that we used the ecological momentary assessment approach, in which participants were monitored for 2 weeks through text messaging. And 3 times a day, we asked them to tell us simply how stressed out they were. And they came to the laboratory twice during the experiment. Once when they endorsed a high level of stress, their own subjective stress, we weren't manipulating anything, and once when they endorsed a low level of stress. And what we find is that there were significant differences between the adolescents and the adult groups, and the adult group we studied, in that when they came to the lab, we asked them to perform another risky decision-making task, the CUPS task, and a cognitive control task. When they came in on the low stress state, based on their own reports of stress, there were no differences in performance on the risky decision-making task between adolescents and adults. But on the high stress state, the adolescents were more likely to make riskier choices than the adults. The imaging data I'm not going to show you because they're messy, and I'm still trying to figure out what it means. But the behavioral data are consistent with what we would predict, that the arousing nature perhaps is stimulating the systems that we think are implicated in risky choice. Okay, so the last thing we wanted to ask was to take a step back, and these manipulations and the way we've examined context in the brain have really kind of been biased towards the idea that context arouses the teenage brain, right? But we were interested in the flexibility of the system, or how malleable it is, in such a way that, if a system is malleable, it should also be responsive in an exaggerated way, and also in a way that dampens the system. It should be responsive in both ways. And so we studied participants within the family context, because there's a long literature suggesting that the family context can really buffer against harmful or maladaptive risk-taking behaviors or internalizing behaviors. And one index of that is something called family obligation values, which is really a way to determine how close a participant feels to their family, or how committed they feel. And we hypothesized, based on work that Eva Telzer, who did this study, had previously shown, that family obligation may decrease the affective and arousing nature of risk-taking, and by simultaneously reducing reward sensitivity and increasing self-control. And so we brought people in teenagers who varied on their level of self-reported family obligation. And again, the question here and the hypothesis was whether or not those who felt closer to their family, whether the family was serving as some sort of buffer to dampen what we think of as a characteristic adolescent striatal response or reward sensitivity. So 48 participants came in and they performed the BART, and they performed a basic cognitive control task, a go, no-go task. And what we find, just behaviorally, is that risk-taking on the BART is decreased in those who show or report greater family obligation. In other words, there's a negative correlation between those variables. And this was also observed and in parallel with activation in this ventral striatum. So this is activation during risk-taking on the BART, which is something we had seen previously and which others have shown as well. But then we regressed it with family obligation. So participants who were more likely to report strong family obligation or closeness to the family showed this decreased response in the ventral striatum. And this is the left and the right striatum. We also found that these participants who showed differences in this family obligation spectrum, those who showed a higher family obligation were more likely to recruit showed better engagement of cognitive control circuitry during the cognitive control task. So, of course, these data are very, very preliminary in answering this type of question, but they do seem to suggest that this family context, this larger difference in a social context, may act on these systems in unique ways. And differ based on the level of family support or closeness. Okay, so together, these data start to suggest that the adolescent brain adapts to the environment in really interesting ways, and there's no one standard adolescent brain, of course. I haven't talked so much about individual differences, but I hope that it's inherent to the data that there are vast individual differences in even within the adolescent sample. Okay, so I will just conclude with some overall statements about how ontogenetic changes in the striatum seem to be associated with unique responses to reward and sensitivity to the environment during adolescence. And I'm really interested in this idea that this heightened sensitivity enhances learning or sensitivity to the environment during adolescence in a way that's advantageous. And that the malleability of the system is probably very critical to changing behavior during this adolescent period that is naturalistic and in ways that is more applied and can be taken advantage of when thinking about interventions or things that would better the well-being of people in this age group. And I'd like to thank all of you for listening. I'm really looking forward to feedback and discussion later. My lab, collaborators, funding, and of course the families who allow us to scan their brain. Thank you. Sorry.
So we have some time for questions.
Yeah.
So I find this very interesting. It seems to me, though, that as an outsider to this field, that there's a significant methodological challenge in identifying how the brain changes from adolescence to adulthood, in that we know that the sample of people who volunteer for these studies is not representative, and this kind of the rules that, or the forces that determine whether an adolescent decides to come to the study could be quite different from the rules that determine whether an adult participates in the study. And so I was wondering if you could talk about how we could gain more assurance that the differences that you're identifying across the lifespan is due to real differences in development versus just differences in the type of people who show up to the study.
It's an excellent question. I think about this a lot. I wish there was a way, I wish I could give you an answer for, the assurance, but I can't. I mean, it's inherent to any type of study where we recruit for either an adolescent imaging study or a college-age sample for behavioral studies. So it's certainly a concern, and we try to be representative in other ways in terms of SES or ethnicity, but really we're just limited by the people who show interest in doing this kind of work, because as you're suggesting, this type of research is not trivial. It's putting them in what some people would perceive as a medical procedure. So we try to be sensitive to that, but it certainly is a concern.
So, yeah, wonderful talk. Thanks. So I'm curious about the last findings in terms of what you think the mechanism might be. So is it the case that family obligation, I mean, it's maybe more than closeness too, right? There's also roles that
that's
the
exactly
children
right.
are playing in the family.
Yes.
So is it a sense, are they sort of more adultified or parentified, and so they're acting like adults because they've been engaged in those kinds of activities or commitments?
That's a really wonderful question, and that's exactly what I think it is, that these kids really differed, and that's the operative difference there is not just closeness, but the obligation to the family or the sense of responsibility. And we asked them extensive questions about what obligation meant. So does that mean you do chores? Do you help with childcare for younger siblings? Make dinner? And all of these things certainly were associated with how much family obligation they felt. And we think that this is because that's in some way strengthening these adult roles, right? Like the process of maturation of the prefrontal cortex isn't just an experience expectant process, but it's certainly based on behavior and exercising the prefrontal cortex, for lack of a better word. So that's what I would hypothesize, that it's really just that taking on the adult roles is playing a role in strengthening them, or at very least changing them. Mm-hmm. Hi, Valerie.
Just brilliant work.
Oh,
I
thank you.
enjoy so much hearing about this, and the way you're bringing together all of these different levels of analysis. And just to follow up, actually, on the previous question, is it possible also, in either direction of influence, by the way, I should say, is interesting, but is it possible that young people who have poor cognitive control, who are higher sensation seekers, are having more family conflict? So in other words, the individual difference in risk-taking may be the source of a lot of conflict with the family during adolescence.
Absolutely, and I think that's that kind of gets at what I started to say about the chicken or the egg, right? They just may be the type of person who's more thrill-seeking in a maladaptive way that would interfere with the otherwise family obligation or relationship that would help buffer it. But I think that's absolutely the case. Mm-hmm. Hi.
I was wondering in the first couple of experiments when you use primary, secondary rewards, whether you have also looked at the cues that predicted, because dopamine shifts with learning from the unconditioned stimulus to the conditioned stimulus, and probably the cues are the main culprits for risky behavior, not the actual reward. And
Yes.
was wondering whether you looked at that as well.
Yes, we have. And actually, when we modeled the data, for the monetary reward, we modeled at the cue, so in anticipation of the reward. And same with the primary reward, the juice. But surprising to us, the effect was stronger at the delivery rather than at the cue predicting the reward. Mm-hmm. But that's another good way to assess learning, right? Learning of that. Mm-hmm. Hi.
So I think for the casual listener, this was really a talk more about go systems than kind of stop systems.
That's right. Mm-hmm.
And probably what we were expecting to hear was a story about increasing ability to regulate, to inhibit, to execute, executive functioning, and all those things that I think we probably think are the hallmark features of the changes in the brain that occur in adolescence. So I was kind of wondering if you would step back a little bit and talk about that balance, and also talk about its implications for thoughts about how to intervene, with the adolescent and what these targets might be.
Certainly. So this is a motivational story, right? But the regulatory story is very similar in that there are changes across adolescence, and the difference there is that the changes show a more linear fashion, that they keep improving, and that improvement has likely to do something with the experience that we were talking about earlier. The individual differences are also very strong. So some kids are better than others at regulating. And, I was recently reading this really interesting hypothesis that perhaps the role of this enhanced motivational, these go systems, their role is really to serve as a punching bag for the prefrontal cortex. In other words, that the way the prefrontal cortex gets hooked up is by having something to regulate. And I thought that was a really interesting idea. I don't know how you would test that, but certainly I think that the regulatory changes that are occurring have a lot to do with the experience of the adolescent. That they interact very nicely with the exposure to cognitive control and just the inherent changes in the prefrontal cortex. Mm-hmm.
A great talk at so many levels, bringing together these different strands. And I think, as Bob was raising, I think the relevance of this to thinking about opportunities for positive behavior change are enormous. I want to push you a little bit more, again, just speculate with this along the lines that Bob was raising. And I think that is that many people imagine that the key to healthier behavior is better cognitive control. But I think what you're raising is that the possibility that positive valuing motivational systems may be at least as important. And I want to go back now to the family obligation story, which is that one possibility of the kids who incidentally, these were determined longitudinally, right?
Yeah.
So some of this addresses Val's question that in Andrew's bigger study, these were kids that the family obligation values were measured prospectively for a long time before they hit
That's right.
adolescence. But the second thing is, one model is, well, they develop really good self-control skills because they're in these roles where they're responsible and they feel that this is important,
right.
valued.
Right.
But the second is the feeling of the trade-offs of taking risks or getting in trouble or doing something wrong has a different meaning at this fast valuing level regarding their family and their role to their family. So the possibility that, not that it's going to be one or the other, but I think we imagine it's about cognitive control. You make yourself not do these bad things,
Right.
but feeling stronger value to do the right things and the
Right.
good things for these reasons could be at least as important. And so when we think about opportunities for positive enduring impact on patterns of behavior, I think the emphasis on cognitive control can obscure what might be at least as important in terms of these motivational systems. I'd be interested in your thoughts.
Exactly, I absolutely agree, and I think that's we're thinking of that these data where it seems that they're less responsive to the only way we can measure risk taking in the scanner. The motivational systems are less responsive in these kids, but that's not because their reward system is broken, right? These are the same kids who showed really robust activation to giving money to their family. Like that's what they find rewarding. And so I think it's really interesting that these systems are sensitive in both domains. So when they're giving to someone else, they show really robust striatal sensitivity, but they're also the kids who show really strong cognitive control and engagement of these systems as well. So it's exactly what you're suggesting. It's not one or the other. It's how these systems are working together and what's valuable to the kid.
Thanks very much. It was all so interesting. And one of the questions that I had, you didn't get to the point of peer affiliation on the list that you had, and I was wondering if you would talk a little bit about your work in that area.
Yeah, I've done less work on that, which is why I didn't talk about it so much. But certainly, several groups are started to ask this question about what the effect of peers are on adolescent brain development. And work you may know by Larry Steinberg shows that when you bring a peer to the lab, it enhances the effects that are normally observed when the teenager is alone. And so one hypothesis is that peers kind of serve as this signal that enhances this inherent response. And Andrew also, and Ron and I have had conversations about how the pro-social nature of that could also be taken advantage of, right? That peers are very motivating and influential to adolescents, and if they can act on these systems, it's not all necessarily, for maladaptive things.
So this was very interesting talk, and by the way, Eva Tenzer is my colleague, so
Great.
we are very happy to have her. I'm interested in the fact that you are at the end of the spectrum of, at the early part of the lifespan, adolescence, and I work on aging, and the dopamine system has been highly implicated in aging, with the idea that if deficiencies in the dopamine system lead to unhealthy aging and cognitive losses. So I'm wondering whether in your thinking you see a continuity between these two, that the kids who develop earlier and have more dopamine, the dopamine system works better at that age, might predict future sort of trajectories in terms of their aging and adulthood.
Oh, that's interesting. I haven't really given that much thought, but I'd be curious to know what your thoughts are as someone who does study aging. Yeah.
I wasn't expecting to. Well, I haven't thought about it much either. I
Yeah.
was just looking at your data
Yeah.
in this context, and I think that's why this conference is so interesting, because it kind of puts people that do very different realms of work together. It's certainly the case that when you look at individual differences and you start looking not just in older adults but in younger adults, you see continuities rather than discontinuities. That is, you see that the same patterns that might be present in aging in a smaller, in a smaller range seem to play out in younger adults. And that's why this is so interesting, because it might suggest the existence of genetics and, in fact, epigenetics motivators. So, for example, the fact that in certain families, kids that take the role of parents have more development of, say, frontal connections, might also be in some way underlined by genetics and not just, or at least environmental promotion of certain genetic development that might be within a family. So there are many, many interesting questions. I think this is an open question, and one that is hard to answer with longitudinal studies, because no one can do a study from one end to the other. Although the nun studies looking, for example, at early writings in nuns and the prediction of those to Alzheimer's seem to have uncovered some of this. So I think this is just an open question.
It's interesting. It would be interesting to know whether dopamine is doing the same thing across the lifespan.
So would either of you think it would be crazy if someone said, oh, I bet you I can learn a lot about how the dopamine system unravels in late life by studying the way it ravels in adolescence? Would you
No, I think it would be a perfectly reasonable thing to do personally. But
Yeah, absolutely.
Right.
So I think this is a wonderful conversation, and I'm smiling at Bob because when he was first trying to engage me in this activity, and I said, I study adolescence, and he says, yeah, but and this is aging, and the idea, these kinds of ideas are exactly, I think, what brought together this network, the BCI network, and I think this is a great topic for the brainstorming session. This is the kind of thing where if the questions are compelling enough, then of course it's going to be hard to do the full longitudinal study, but maybe there are animal models, maybe there are longitudinal studies where you can go back and with the right focus on the right question in adolescence, if not the writing sample, other things like that. I think this is exactly the kind of thing that we think could be very exciting to lead to innovative approaches to ask questions that wouldn't normally emerge unless you get this interdisciplinary group looking at two different inflection points.
Hello, my name is Susan Hoffman. I'm the director of the Osher Lifelong Learning Institute,
Hi.
which is doing research on the older adult learner. And particularly what we've been spending time with over the last 2 years is the over 80 learner. And what we're encountering is the environmental barrier for most of them around the ability to hear. And what we also know from the research, not ours, that there is a direct link between those who cannot hear, their withdrawal from society and from public world, the increase of depression, and an earlier onset of dementia. So I think that as much as I appreciate biochemistry and all the things that we are learning, through MRIs about what is happening over the lifetime, I think let's not see that there are things that we might do that would significantly shift altering depression, social engagement, ongoing learning for those that are in later life.
Absolutely agree. That's so fascinating. That's something that may easily be missed, right? Without studying it, would explains what it sounds a lot of the data.
Thank you. Enjoyed this very much. I wanted to ask you, my research area is alcohol and drug abuse. Could you speak to what you can pick a drug, recreational drug, would do to these regulatory processes, particularly one that might affect the dopaminergic system?
You would think that there would be more data on how drugs of abuse influence the adolescent brain. Most of the work has been conducted with marijuana. It's the drug of choice, alcohol too. And what most of these data from an imaging perspective have looked at basic cognitive functions like response inhibition or working memory. And there seems to be an association with those who are heavier users show deficits in these cognitive behaviors and also in the systems that support it. But I have to say the data are not as strong as we would predict. And I think it's because samples are limited and it's challenging to do these types of studies because of the question that came up earlier that they are self-selected groups. So it's hard to get pot users to commit to to coming to the lab and staying still, all these things.
Area of expertise, I know, but I wanted to. So one of the other interesting things about adolescence to me, which is a kind of the opposite of in late life, is sulkiness. Do you understand that? Can we understand that at all in terms of the maturation of these circuits? Where does that come from, or should we just think about it as purely contextual?
Do you mean sulkiness in a clinical way, like mood disorder? Do you just mean
No, that's sort of
brattiness?
irritating brattiness
Yeah.
that defines this period of life.
I don't know what that's about. I can't speak to that. But if you're thinking clinically, there are data from Ron's group also in Pittsburgh suggesting that teenagers who show these dampened affect or dampened hedonic experience do show dampened responses in these systems. But the sulkiness, I don't know.
All right, thanks for
Thank you.
talking. And